This is Lecture 3 for Microbiology. These notes are going to correspond to the Bellman Chapter 3 text, and they will correspond to Chapter 4 in the tutorial text. We're going to be looking at cell structures and how cells function. When we look at cells, we need to define life. There is no magic ingredient that makes things alive, so instead we define properties of life. Things that are alive are going to have growth, they're going to increase in size. They'll reproduce, either reproducing themselves sexually or asexually. They have responsiveness, where they're going to respond to changes in their environment, and they will undergo metabolism. They'll take in and use nutrients. Metabolism is looking at all of those chemical reactions that go on in a cell. We have two main types of cells, eukaryotes and prokaryotes. Prokaryotes do not have a nucleus. There's no nuclear membrane-bound genetic material. They're going to include the bacteria and the archaea. Eukaryotes are going to have membrane-bound DNA in their nucleus. They will also have organelles, which will be membrane-bound specialized structures. Prokaryotic cells can take on characteristic shapes and arrangements. The coccus or cocci shape is round. The bacillus is rod-shaped. A diplococci or diplobacilli are going to remain in pairs after dividing, so the cells will be seen in pairs. Streptococci or streptobacilli are going to remain in chains after dividing, so these cells will be in chains. Tetrads are going to be when they form groups of 4. Sarcinae will be in cube-like groups of 8. Staphylococci will be in grape-like clusters. The coccobacilli are oval-shaped rods. They almost look like cocci. These can be kind of hard to distinguish underneath the microscope. Vibrios are going to be curved rods. The spirilla or spirillum are going to be helical shaped like a corkscrew, but they're going to have rigid bodies. The spirochetes are going to be helical shaped and flexible. When we look at the prokaryotic cells, the glycocalyx is sometimes referred to as the sugar cup, the sticky substance made up of polysaccharide and or polypeptides that surround the outside of the cell. The capsule is called the glycocalyx in the prokaryote. It's made of organized repeating units of organic chemicals that attach to the surface. These help to protect the cell from desiccation. It may also protect the host because they're going to be chemically similar to the host. The slime layer is the loose water-soluble glycocalyx. This protects from desiccation and allows it to attach to the host or to surfaces as biofilms. EPS, or extra polymeric substance, is the glycocalyx that helps cells and biofilms attach and facilitates communication between the cells. A flagella is a whip-like tail for motility. It's a long filament. In bacteria, they rotate 360 degrees like a motor. They're 20 nanometers in diameter and made of flagellin protein around a hollow core. The base of the filament inserts into a hook, and then the basal body anchors the hook and filament to the cell wall. Here you can see what the structure looks like for the prokaryotic flagella versus a eukaryotic flagella. So they do have a different structure and a different movement. The prokaryotic flagella is going to spin where the eukaryotic one will move in waves. There is a little video here on bacterial flagella that I would encourage you to watch outside of this lecture. Remember, I can't access these videos in the lecture without YouTube flagging them. We can look at the arrangement of the flagella. Monotrichous are just a single. Lophotrichous are going to have a tuft of flagella at one end. The amphitrichous are going to be flagella at both ends, and peritrichous has flagella covering the surface of the cell. A polar flagella is found just at one end. The endoflagella are a little bit different. These flagella spile tightly around the cell in spirochetes, and they form an axial filament that wraps around the cell between the cell membrane and outer membrane. The rotation of the endoflagella causes the corkscrew rotation you see. These are found in the Treponema pallidum and Borrelia burgdorferi. Here's a video that shows the spirochetes. Motility means that the organism can move by itself. It can move towards a stimulus. We call this taxis. Some examples would be chemotaxis when it moves towards a chemical signal, or phototaxis when it moves towards light. Pili are little tubules composed of the protein pilin. They're shorter than flagella but longer than fimbriae, and they're used to move across a substrate or in conjugation for genetic transfer. They can create the smooth gliding motility we see in the Mycoplasma. Fimbriae are non-modal extensions that we see in gram-negative bacteria. This allows them to adhere to one another and to other substances. These are important in the biofilms, which are those slimy masses of bacteria that adhere to a substrate. Some of them will carry enzymes. The cell walls are going to have peptidoglycan in them. Peptidoglycan is made of two regularly alternating sugars, abbreviated as NAG and NAM, N-acetylglucosamine and N-acetylmuramic acid. These are attached to amino acid phosphorages. These are some pretty nice pictures here of the differences in the cell walls. A gram-positive cell wall is going to have thick peptidoglycan with lipoteichoic acids that will anchor the peptidoglycan to the cell wall and membrane. These will retain the crystal violet stain, so they will appear purple underneath the microscope. Some have mycolic acid to help resist desiccation. These are more difficult to stain because it's usually going to be acid-fast. This would include the Mycobacterium tuberculosis, and there are other stains we can use for these. The gram-negative cell wall has a thin layer of peptidoglycan. They have a bilayer membrane outside with porins, which are channel proteins, and LPS. LPS, or lipopolysaccharide, is a sugar and a lipid. The lipid portion is known as lipid A, or endotoxin. It's released from dead cell walls when the wall breaks down. This is important in looking at disease because it will trigger fever, inflammation, shock, and clotting in humans. The core polysaccharides attach to a lipid and provide stability. O polysaccharide extends outward, and it functions as an antigen. Your periplasmic space is going to be the space between the cell membrane and the outer membrane in a gram-negative cell. This will contain water, nutrients, digestive enzymes, and proteins. There are a few bacteria that don't have cell walls. This includes the Mycoplasma Pneumoniae. A protoplast is what you would have if the cell wall is damaged or destroyed, but the cellular content remains surrounded by the plasma membrane. They're usually spherical, and they can carry out metabolism. L forms, we see these in Proteus and some of the other, where in general they can lose the cell wall in response to things like penicillin or lysozyme, and they will swell into these irregular shaped cells called the L forms. These can live and divide without returning to a walled state. The spiroblast occurs when lysozyme is applied to a gram-negative cell wall. It's not destroyed as much as it is in a gram-positive cell, so they'll form the spherical structure. The protoblasts and spiroblasts can be ruptured by osmotic lysis, so they're not as durable as the regular cell. Acid-fast cell walls have high concentrations of hydrophobic wax called mycolic acid. They won't gram stain, but they can be stained with carbofusion, which penetrates the cell wall when heated. The archaea cell walls don't have any peptidoglycan. They have a variety of special proteins and polysaccharides. They will gram stain purple and pink. The cytoplasmic membrane is going to be your cell membrane or plasma membrane. It's a phospholipid bilayer. It's made up of two layers. One end is polar, and then you have one that is nonpolar on the phospholipid. The middle layer is going to be hydrophobic, and the surface is hydrophilic. So you'll have two layers facing each other in opposing directions with the polar ends on the outside and the nonpolar tails facing towards the middle. The cytoplasmic membrane, we use a fluid mosaic model to describe it. The proteins are arranged like tiles, and they're free to flow laterally within the membrane. These membranes have selective permeability, so some substances can pass while others do not. The chromatophores are infoldings in the plasma membrane with enzymes and pigments for photosynthesis. Glycoproteins and glycolipids on the plasma membrane will help to lubricate the cell, and they're involved in the cell-to-cell interactions. This is a video clip here that shows the fluid mosaic action. of the phospholipid bilayer. In the cells, we have a difference in concentration that can cause your electrochemical gradient. The gradient is driven by a difference in the concentration on the two sides of the membrane. The electrical portion is going to be the voltage across the membrane, and the electrochemical gradient is the electrical chemical gradients together. By having different charges on the sides of the membrane, that will create the potential to do work that we can measure as the voltage. With passive diffusion, you have movement down the concentration gradient. No energy is required. For this process, it's completely passive. Facilitated diffusion is going to need help or something to facilitate it. It needs a pathway for diffusion. It's usually an integral protein. Osmosis involves the diffusion of water across a selectively permeable membrane. Something that is hypertonic is going to have higher solutes. Hypotonic is going to have lower solutes, and isotonic will have the same solutes on both sides. Active transport uses carrier proteins. This will require ATP because it is an active process. Here you'll have gates that will act as channels or pores that are controlled. A uniport is going to move one substance at a time, while an antiport moves two chemicals in opposite directions. A symport is going to move two chemicals together. In group translocation, this is an active process and only seen in some prokaryotes. The substance is transported, but as it's transported, it's going to be chemically changed during the transport process. Here we have a short little video clip on cellular transport. The cytoplasm is going to include your cytosol, which is the fluid portion, inclusions, ribosomes, and the cytoskeleton. The nucleoid is the region of the prokaryote that contains the DNA. Plasmids are small, circular, double-stranded DNA molecules. They're not connected to the main chromosome, and they will replicate independently. Inclusions are deposits of lipid, starch, or other compounds containing nitrogen, phosphate, or sulfur, or magnetite. We see the magnetite in the Magnetobacteria, gas vesicles in many of the aquatic cyanobacteria. Carboxysomes are going to carry enzymes for carbon fixation. The metachromatic granules are large inclusions that will stain red with certain blue dyes like methylene blue. Collectively, these are called gluten, and they represent a reserve of inorganic phosphate. They can be used in ATP synthesis. Polysaccharide granules, these will include glycogen and starch granules. Glycogen stains reddish-brown with iodine, where starch will stain blue with iodine. Ribosomes are used in protein synthesis. They are a little bit different in the prokaryotes and eukaryotes. Prokaryotes have 70S ribosomes. Eukaryotes have 80S ribosomes. The S is for Svedberg's, which is a measure of the sedimentation rate. The prokaryotes have 30S and 50S subunits and are composed of ribosomal RNA and protein. Many antibiotics will actually act on the 70S ribosome and don't harm the 80S ribosomes. The cytoskeleton is an internal network of fibers. This helps to establish the cell's basic shape. These are lacking in the sphere-shaped prokaryotes, and they are simple in the rod-shaped prokaryotes. Endospores are going to be dormant entities. Some examples are the Clostridia and Bacillus. These are a defensive strategy used during unfavorable conditions. They're resistant to drying, heat, radiation, and lethal chemicals. They germinate again when the conditions improve. The Archaebacteria are similar to the bacteria. When we look at some of their structures, some of them have a hamus or a hami, which is a proteinaceous appendage used to attach to surfaces. Most archaea are going to have cell walls, but they're different than bacterial cell walls. They lack peptidoglycan. Gram-positive archaea cell walls are thick and stained purple. The gram-negative archaea cell walls stained pink, and they have an outer layer of protein instead of a lipid bilayer. The archaeribosomes are 70S like the bacteria, but they have different proteins. They'll use different enzymes to make RNA and code more similar to the eukaryotes. Now we'll look at some of the external structures of the eukaryotes. The glycocalyces is a sticky portion that helps to anchor the cells, provides strength and protection. It's found in animal cells but not plant cells. It's not present in the cells that have walls. Flagella are different in the eukaryotes than the prokaryotes. They're going to be made of tubulin protein in chains to make microtubules. You have 9 pairs of microtubules around 2 microtubules in the filament, so we refer to it as a 9+2 filament. There's no hook, and the basal body has triplets of microtubules with no microtubule in the center, so we refer to it as a 9+0 basal body. Cilia are only present in eukaryotes. They're not present in prokaryotes. They're a hair-like structure. They're going to have a 9+2 shaft and 9+0 structure in the basal bodies. Looking at the eukaryotes, when we compare them to the prokaryotes, their cytoplasmic membrane is going to be a fluid mosaic like the prokaryote, but it is going to contain steroids that help to maintain the fluidity. Both control movement in and out of the cell. There is no group translocation in eukaryotic cells. They have membranous rafts, which are distinct assemblages of protein and lipid that stay together as a functional group. Some of the viruses will actually use the rafts to enter the cells. The eukaryotic glycocalyx is a sticky layer that's anchored to the cell membrane. This anchors the cell, protects it, is involved with cell-to-cell recognition and communication. It's found in animal cells and most protozoa, but not in cell walls, cells with walls like the plants. Endocytosis and exocytosis are ways of getting larger particles in and out of a cell. With endocytosis, the membrane engulfs the particle instead of using group translocation. This is done by manipulating the membrane around the cytoskeleton. With exocytosis in eukaryotes, it's a reverse of endocytosis. This is used to export a substance. Here we have a little video clip of endocytosis. Eukaryotic ribosomes are larger. They're 80S with 60S and 40S subunits, and they may be attached to the ER or endoplasmic reticulum. The cytoskeleton is extensive. You're going to have microtubules of tubulin, microfilaments of actin, which are smaller, and then the intermediate filaments made of various proteins. This will allow for cytoplasmic streaming, where you have movement of the eukaryotic cytoplasm. This helps to distribute the nutrients and move the cell. Your centrioles or centromere lie at right angles near the centrosome. The centrioles have a 9+0 arrangement. They're part of cytokinesis. They help to form cilia and flagella. Their function is not fully understood at this point. With the nucleus, you can have single or multiple. The plural term is nuclei. This will contain most of the genetic information in the nucleoplasm, which is a semi-liquid matrix. The nucleoli is a plural for multiple nucleus. Nucleoli is going to be the region where the RNA and ribosomes are synthesized in the nucleus. Chromatin is a thread-like mass of DNA. It's going to contain proteins called histones for packaging the nuclear DNA. This is the loosely coiled format that you're going to see the DNA in most of the time. Your nuclear envelope will have nuclear pores that will allow things to go in and out. It is a double membrane made up of phospholipid bilayer, and those nucleopores control what goes in and out. Your eukaryotic flagella are structurally and functionally different than in prokaryotes. The cytoplasmic membrane surrounds the flagellum. They're composed of tubulin in a 9+2 pattern, anchored by a basal body, which is 9+0. There's no hook, and they undulate in a wave-like motion rather than rotate, and you usually only have them at one pole. endoplasmic reticulum, or ER, is continuous with the outer membrane of the nuclear membrane. It's made up of hollow tubules in its arrangement, and it's involved with transport of packaging. Smooth ER does not have ribosomes on the surface. It's more involved with lipid synthesis and transport. Rough ER, or RER, has ribosomes on the surface. The proteins made by the ribosomes are inserted into the lumen of the ER and transported. Your Golgi apparatus is going to receive, process, and package large molecules. This will get them ready to export them from the cell in sacs called secretory vesicles. The secretory vesicles are these secretions that will fuse with the cytoplasmic membrane and do exocytosis. A vesicle or a vacuole is a large sac that's used to store or transfer chemicals. We see large vacuoles in plants and algae to store starch, lipids, and other substances. Lysosomes are found in animal cells. These contain catabolic enzymes. Peroxisomes are going to be vesicles that have oxidase and catalase, as well as other enzymes to degrade poisonous metabolic wastes. These are particularly abundant in liver and kidney cells. Mitochondria are involved with ATP production. This is going to have an inner membrane called the cristae that is going to have two phospholipid bilayers. The inner matrix will contain small prokaryotic 70S ribosomes and circular DNA. The chloroplasts are your light harvesting organelles in the photosynthetic eukaryotes. They have two phospholipid bilayer membranes, 70S ribosomes. The photosynthetic prokaryotes have photosynthetic lamellae instead. The Endosymbiotic Theory is going to explain how eukaryotic cells formed from the phagocytosis of smaller aerobic prokaryotes. So we had aerobic prokaryotes that ended up being engulfed by larger anaerobic prokaryotes. The smaller ones became the internal parasite and eventually lost their ability to exist independently, but they did retain some of the portion of their DNA, the ribosomes, and the cytoplasmic membrane. This explains why the mitochondria and the chloroplasts are going to have 70S ribosomes. Your photosynthetic eukaryotes were actually going to be developed in a similar fashion. They engulfed a photosynthetic prokaryote that lost its ability to survive on its own. Here's a quick little animation on the endosymbiotic theory.